Welcome, students, to today's histology lecture. In today's session, we will delve into the fascinating field of bone development and remodeling. We will explore the intricate molecular mechanisms, cellular processes, and genetic determinants that contribute to the formation and maintenance of healthy bones. Furthermore, we will examine the impact of various external and internal factors on bone development, including mechanical forces, systemic diseases, and immune cell interactions. Without further ado, let us begin our journey into the captivating world of bone histology. Bone development and remodeling rely on WNT signaling, a fundamental pathway in regulating osteoblast differentiation and bone formation. WNT signaling includes two branches, canonical and non-canonical pathways. The canonical pathway involves beta-catenin activation, while the non-canonical pathways impact cell polarization and calcium signaling, indirectly affecting osteoblast differentiation. A balanced interaction between these pathways is vital for maintaining bone mass and integrity. Transcription factors like RUNX2, Asterix, and ATF4, alongside signaling pathways like TGF-beta and BMP, regulate osteoblast differentiation and function. Their coordinated activities control bone formation and remodeling, making them crucial in understanding bone development and pathology. Endochondral ossification is the process by which long bones are formed. Genes like SOX9 and I are important for regulating cartilage formation and chondrocyte differentiation. Disruptions in these genes can lead to skeletal abnormalities. Growth factors also play a role in controlling the process. Mechanical forces, such as strain and fluid flow, affect bone modeling and remodeling. Osteocytes sense and transmit signals to osteoblasts and osteoclasts, causing bone remodeling. Fluid flow induces shear stress on osteocytes, triggering intracellular signaling and promoting bone remodeling. Understanding these processes is important for bone health. Osteocytes are vital cells in bone development and maintenance. They sense mechanical stimuli, regulate mineral balance, and contribute to bone remodeling. They release enzymes for matrix degradation and regulate the differentiation of other bone cells. This knowledge aids in understanding bone health and potential treatment strategies for related conditions. In summary, during cartilage development, chondrocytes undergo proliferation and hypertrophy. Chondrocyte proliferation ensures the formation of a proper cartilage template, while hypertrophy leads to an increase in size and the expression of genes involved in bone formation. Several signaling pathways regulate this process. Understanding cartilage development is essential for understanding skeletal disorders. Systemic diseases like diabetes and hormonal disorders impact bone development and integrity. In diabetes, high blood glucose levels form ages that affect bone structure and osteoblast function. Insulin resistance hinders bone remodeling, reducing formation and increasing resorption. Hormonal disorders, like hyperparathyroidism and hyperthyroidism, accelerate bone turnover, causing bone loss and fractures. Understanding these effects is crucial for managing bone health in patients with these conditions. Bone mineralization and matrix formation are intricately regulated processes involving the deposition of calcium and phosphate ions onto the collagen matrix. Various molecules and proteins participate in orchestrating bone mineralization. One critical protein involved in bone mineralization is osteocalcin. Osteocalcin is synthesized and secreted by osteoblasts and is known to bind calcium ions, facilitating their deposition onto the collagen matrix. Another protein, osteopontin, plays a role in nucleating hydroxyapatite crystals, the mineral components of bone. In addition to proteins, small molecules, such as pyrophosphate, P, are crucial regulators of bone mineralization. P is synthesized and released by osteoblasts and inhibits hydroxyapatite crystal growth. Its presence ensures that mineralization occurs in a controlled and organized manner. The precise coordination between various molecular players ensures the formation of a highly organized and mineralized bone matrix, which provides the necessary mechanical strength and support for the body. Angiogenesis, or the formation of new blood vessels, is important for bone development and fracture repair. Blood vessels provide oxygen, nutrients, and essential factors for bone cells to function properly. Angiogenic factors like VEGF promote the growth of new blood vessels, aiding in the healing process. Epigenetics, through DNA methylation, histone modifications, and non-coding RNA molecules, influences gene expression during bone development. Methylation patterns determine lineage commitment, while histone modifications regulate osteoblast and osteoclast gene expression. Non-coding RNAs, like microRNAs, play a role in bone biology. Manipulating epigenetics can improve bone conditions. Developmental diseases, such as dwarfism and skeletal dysplasias, have a profound impact on bone development and growth. These conditions are often characterized by abnormal skeletal proportions, bone deformities, and impaired linear growth. One example of a developmental disease affecting bone development is achondroplasia, the most common form of dwarfism. Achondroplasia is caused by a mutation in the FGFR3 gene, leading to disturbed endochondral ossification and impaired longitudinal bone growth. Individuals with achondroplasia typically exhibit short stature, disproportionate limb lengths, and abnormalities in the spine and skull. Skeletal dysplasias encompass a broad spectrum of congenital disorders affecting bone and cartilage development. These conditions can affect various aspects of bone growth, including chondrocyte proliferation, differentiation, and matrix synthesis. Understanding the impact of developmental diseases on bone development provides insights into the underlying molecular and cellular mechanisms and paves the way for improved management and treatment options for affected individuals. Immune cells like macrophages and regulatory T cells are important in bone development and remodeling. Macrophages help form bone-resorbing osteoclasts, while TREGs secrete anti-inflammatory cytokines that inhibit bone resorption. Imbalances in TREG function can lead to excessive bone loss. Other immune cells also contribute to the regulation of bone remodeling. This knowledge can help develop new therapies. In summary, bone development and remodeling are complex processes involving molecular regulation, cellular interactions, and external influences. Understanding these mechanisms helps diagnose and treat skeletal disorders for an improved quality of life. Explore further to uncover more about bone histology. Thank you for your attention.